thanks Keith and Hans-Peter for inviting me again. This is one of my most favourite meetings. It's really fantastic. And I'm actually really glad I followed Laurie, because my talk is very translational, and I come to this as a bone marrow transplant doctor, and so 20 years experience of giving virus-specific T cells to people with cancer or who are having a bone marrow transplant for a deadly disease, is quite different to, like you just heard, if someone's taking a pill a day and feeling completely well. How do you, as a responsible doctor, consent patients to first-in-human therapies? So I don't have the answers to that, but I do have what we're doing in this space. MAC. Okay. I do have some disclosures in the antigen-specific T cell field, but not so much in the HIV field. So this is my summary for the community. You've heard a lot today, and you will hear more about gene-engineered cells, especially in the T cell therapy field, about CAR T cells or chimeric antigen receptor T cells. So my talk's really focused today on antigen-specific T cells, and they are not CAR T cells, so if you just remember that, that's great. Antigen-specific T cells have been highly effective and safe for people with EBV-associated cancers and for life-threatening virus infections after bone marrow transplant, with greater than 90% efficacy with toxicity rates of 1.2%. So the question now is, will HIV-specific T cell therapy be safe and effective as a cure strategy for HIV? So just to highlight the different T cell therapies we're talking about, so there's the CAR T cell therapies, and I know Jim's going to talk in detail about that next, so I won't belabor the point that this is where you gene engineer a T cell with a retroviral or lentiviral construct which expresses your CAR, which is basically a single chain variable region of a monoclonal antibody engrafted through a hinge spacer domain onto usually a co-stimulatory moiety and the T cell receptor. So now the engineered T cell is able to recognize its target like an antibody, but kill it like a T cell. So in contrast, antigen-specific T cells are like what we're so used to in bone marrow transplant, the donor lymphocyte infusions, but instead we're actually pulling out your antigen-specific T cells or priming the antigen-specific T cells, not gene-engineered, by pulling out these physiologic T cells that are able to recognize and kill multiple different virus-associated antigens on cell surfaces. And then just to complete the story are TCR transduced T cells, where you actually can build on the antigen-specific T cell platform by pulling out a T cell receptor for a single epitope and gene engineering a T cell to recognize a single viral epitope. So we recently published a review article on the T cell therapy field for HIV, and really the pioneering work of Phil Greenberg here using CD8 clones really showed the need that this lack of CD4 help really limited the persistence of these cells in vivo. Furthermore, targeting single epitopes really put the system, if you like, under stress of immune escape with antigen or epitope loss. So we tried to overcome these limitations by building on the platform that we'd used to generate EBV-specific T cells and CMV-specific T cells for patients post-bone marrow transplant. So essentially, we took dendritic cells. This is from HIV-positive individuals, and we pulsed them with HIV peptides for GAG, POL, and NEF. So they were basically, they were a proprietary algorithm that we got from JPT. They're 15 amino acids long, overlapping by 11 amino acids, and in the presence of cytokines, you stimulate and expand an HIV-specific T cell population. We called this product HXTCs. We used artificial antigen-presenting cells, K562-based cells, to expand the cells to adequate numbers to infuse. So this was collaboration done with David Margolis at UNC, and this was a graduate student of mine at the time, Sharon Lamb, who showed that from HIV positive donors, irrespective of their HIV history, you could expand GAG, POL, and NEF-specific T cells from these individuals within 2 to 3 weeks expansion time. And she showed here, using laboratory strain of HIV, that you could have suppression of CD4 cells infected with a laboratory strain of HIV when you mix them with the autologous HXTCs. And in a subsequent article by Julia Sung from David's group, they showed that you could see the same results with the autologous virus, so suppressing CD4s infected with the patient's autologous virus. So we took this to the clinic, and this was the initial study. It was a fixed dose study, 2 times 10 to the 7 per meter squared per dose, 2 infusions, 2 weeks apart. We treated a total of 8 participants, and there were no severe adverse events and no toxicities related to HXTCs. So please note there was no lymphodepletion used. These cells were just literally infused into the participants in the outpatient clinic. So this paper was published in the middle of last year and suggested that the HXTCs alone may confer a measurable augmentation of the HIV-specific T cell response in vivo, but it really was unclear if it had a role as a single agent as a cure strategy. So we now have a combined protocol that's ongoing, and so we're combining it with David's LRA that was du jour, several years ago, I guess, so vorinostat. And so currently we've treated 4 participants with HXTCs plus vorinostat, and we're currently analysing that data at the present time. So I think, where we're going, we're doing these things all and these trials concurrently. Another way to sort of overcome the limitations of the HXTCs alone would be to target just the conserved epitopes of HIV, GAG, and POL. So as part of the BELIEVE Collaboratory in collaboration with Brad Jones from Cornell, we've developed a new product, the HST-NEAT product. And so here for GAG and POL, we're using the peptide panel based on the conserved mosaic vaccine of Hankey and Korber, which probably I don't need to tell this audience is 80 to 90% perfectly matched to all M viruses. So we're using the same NEF pep mix that we used for the HXTC protocol. So now we have a GAG, POL, and NEF specific T cell product again, but hopefully this time the product only recognizes conserved sequences of GAG and POL. In vitro, these cells perform similarly in terms of global specificity. And interestingly, when we do epitope mapping for GAG, it appears that you have very similar epitope-specific repertoire. This is all from HIV-positive donors. But for POL, you can see a much broader response to POL epitopes with our HST-NEAT product in blue there. So what does this mean in vivo? So we started initially with a murine model. This was developed by Brad. He basically infects CD4 cells with a laboratory strain of HIV, as well as obviously these are from HIV-positive participants, and then engrafts the mice with the CD4 cells, and then we give our T cell product on top of that. And so he's tested HXTCs at the top, HST-NEATs in the green, and other types of clones. I won't get into the weeds. Sorry, the middle is this is the HXTCs, and this is the HST-NEATs. And this is data he sent to me a while back, but actually recent data is holding true that it might be donor dependent, but HST-NEATs do seem to better clear the viral load in this mouse model than the HXTCs do, suggesting that these guys undergo immune escape after maybe initial decrease in viral load, and these guys can continue to clear the viral load. So that's sort of encouraging as we move now to the clinic. So this study is open and enrolling, so it's actually very much the same as the HXTC protocol. Initially, it's a safety trial alone, just with the product alone. This time it's in collaboration with the Whitman Walker Clinic, and so they have selected participants who have where we are able to measure the reservoir, so have a reasonable IUPM number ahead of time. So this is the setup here, same dose as we did for the HXTCs, again without lymphodepletion, giving these cells 2 weeks apart. Initially, actually, the FDA said we have to treat the first 3 participants, giving these cells 4 weeks apart, and then we can do 2 weeks apart for the subsequent 3 participants. So we treated our first patient last month, and we're generating another product on participant number 2. So this is ongoing, and the idea is that we'll establish the safety of the HST NEAT product alone, and then work on doing combination strategies. So so that's where we're doing within the BELIEVE Collaboratory, but what can we do at the same time to really enhance persistence and overcome resistance, especially in the bone marrow transplant setting, given my area of expertise? And so one area is obviously lymphodepletion, and I know Jim's going to talk to you about how he does lymphodeplete his participants pre-CAR T cells, but the FDA wanted us for this first-in-man product or first-in-human product to not do that. So the only setting we felt we could do this ethically in was in the transplant setting, where HIV-positive individuals were needing a transplant for an underlying cancer or other life-threatening disease. So we have 2 strategies ongoing. One is funded through the BMT Clinical Trials Network in collaboration with Richard Ambinder from Hopkins and Helen Heslop from Baylor. So this is building on the study that was done by the CTN and the AIDS Malignancy Consortium, showing that individuals with HIV actually have the same outcome in terms of lymphoma outcome as HIV-negative patients after autologous bone marrow transplant. But for the HIV-positive individuals, while their HIV levels did go to undetectable, all remained on antiretroviral therapy. So no one was advocating this was a cure strategy alone. So we're proposing to build on that autologous transplant study and then use the autologous HST NEATs in the lymphopenic period after autologous stem cell transplant. And so the endpoint is really an HIV endpoint, looking at reduction in intact proviral load. So this is, we're also hoping that we can see a reduction in the intact proviral load by 70% at 6 and 12 months. So this is this study's design. We standard conditioning regimen for lymphoma patients, pre-autologous stem cell rescue here, then give the autologous HIV-specific T cells early, so within 3 to 5 days post-transplant, and then monitor not only for their lymphoma, but HIV outcomes at different time points, as shown here. So that's sort of the lymphodepletion question that we're hoping we can answer, and we will see how that goes in a national study. But then last time I was here, actually Timothy Brown was the only person cured of HIV post-allogeneic stem cell transplant, but now it does appear that the London patient has also had undetectable virus for over a year off antiretroviral therapies. And the main difference and significance here is that Timothy Brown received a fully ablative conditioning regimen, and he also had to have 2 bone marrow transplants, so he had heavy preconditioning chemotherapy. Whereas the London patient received submyoablative chemotherapy regimen, so-called mini transplant, which has, it's not toxic, but it has much less toxicity than a full conditioning regimen. And then both received homozygote donors for the CCR5 delta 32 mutation, which, as you know, is only 1% of the white population. So we're trying to see if we can capitalize on this post-allo transplant setting by growing HIV-specific T cells from delta 32 homozygote donors. So where are these magic donors? So there are about 5,000 in the cord blood transplant registries in USA and Europe. And there's about 20,000 donors in the National Marrow Donor Unrelated Donor Registry in Germany. So our plan is, and this is through a PO1 I have with Rich and Binder and Rick Jones at Hopkins, is to administer the CCR5 delta 32 donor-derived HIV-specific T cells to HIV-positive individuals who are receiving an allo transplant for an underlying malignancy from the same delta 32 donor. And because Hopkins have pioneered the use of the haplo donor transplant, we can prioritize the CCR5 delta 32 status over HLA typing because we can get away with only a 3-on-6 match. So we had to first show that we could grow these HIV-specific T cells from seronegative donors, and this was work done by Shabnam Patel. We basically used the same platform as we did for the HIV-positive donors, which is very different to what we did when we are generating naive CMV T cells from naive donors. So anyway, this is the same setup that we did for the HIV-positive donors. And you can see here's adult donors and cord blood donors. You can see nice central memory phenotype in about 30% of the products, which we know is important for long-term persistence. And unlike the HIV-positive donors, we actually do see more CD4 positive HIV-specific T cells in the final products. We know that from the seronegative adult donors that these cells really are derived from the naive compartment using these sorting experiments for CCR7, CD62L starting populations. Obviously, cord blood is inherently naive, so you can see nice GAG, POL, and NEF-specific T cells in these final products that are derived from cord blood donors. And that furthermore, in vitro, these cells are able to suppress CD4 T cells infected with laboratory strain of HIV. So this is in the adult donor population, and this is in the cord blood donor population. So we're now working on replicating these results from these precious CCR5 delta 32 homozygous donors, and so far we can see we can generate these GAG, POL, and NEF specific T cells from these seronegative delta 32 donors, homozygote donors. So this is where we're at right now, where the allo transplant trial is written and is currently at the scientific review committee at Hopkins. And so basically we'll see if we can affect any impact on the reservoir post-allo transplant with this adoptive transfer of the seronegative donor HIV T cells early after transplant. But I'm not saying no to gene editing strategies, and we do have 3 key gene editing strategies that we're exploring for these products, and I'd just like to do a shout out for Ping Shen, who's here, who's working on CRISPR-Cas9 strategies for to knock down different genes to render these seronegative donor HIV T cells resistant to HIV. So these are my take-home slides. So the first one's for the community. Again, just to reiterate that antigen-specific T cells are not CAR T cells. So far, the HIV T cells that we've administered to HIV-positive participants have shown an excellent safety profile. I do believe that safety is critical for broadening the applicability of this approach to effect a cure. I think it is a highly exciting strategy, but will have to be used in some sort of combination setting, whether it's with latency reversing agents or in the post-transplant setting. So these are my concluding remarks for the whole community. I think that the successful use of HIV-specific T cells in the autologous setting will need to be done in combination, and in the post-allo setting may offer a curative strategy, especially if additional gene editing strategies are employed as well. So I tried to acknowledge everyone along the way. This is years of work, so thank you very much. But I would like to can I just say a shout out to our cab? So Richard, I see you're here, so thank you, Richard. He's not even looking at me. And I don't know if Rico's here, but I want to thank, because obviously we can't do this without you, so thank you.
Thanks a lot, Kat. So let's do 2 questions. The questions? Okay.
I see that you use a combination of cytokines for the expansion. Have you looked at all at comparing it with CD3, CD28 expansion? Is there some advantage, disadvantage of doing it that way?
So when we do it that way, we lose specificity. So it is sort of a magic combination, if you like, of the cytokines we use for the priming or the initial stimulation versus the expansion. But if you just use the CD328 post the first stimulation, you will lose specificity of your product. Yeah.
Do you know why that is?
I think it's because it's such a powerful stimulus that it's pulling out, the non-specific T cells at the same time. So you haven't given the system time to be enriched enough for your antigen-specific T cell population.
Thank you.
Maybe I missed it, but from your past and sort of ongoing studies, what % of the infused T cells are CD4-specific HIV T cells, and what's their fate?
So in the HIV-positive individuals, the percentage is probably in the order of 10 to 15%. In the seronegative donors, we're at 30 to 40%. And so, yeah, and that's what we see, honestly, in CMV when you're dealing with CMV positive versus negative donors.
But what's the fate post-infusion? How long do they last?
Remembering we haven't given these in the lymphodepletion setting yet. So in the lymphoreplete host, it seems like they're persisting a maximum of 6 to 12 weeks.
Okay.
So yeah.
I think we have to move on. Thanks so much.
Thank you.